Greetings, everyone. I hope you can hear me well, and welcome to today's webinar. I'm Kübra. I'm an Assistant Marketing Manager of the International Business Division here at IVIM Technology. And I'm delighted to have you all here today. With this webinar, we will delve into in vivo cellular level imaging of internal organs in live animals for immunotherapy and cancer research. So let me introduce our guide for today, Hayden Won. She has a master's in forensic science and has previously worked in one of the leading institutions in Korea, which is Seoul National University Hospital. So she is currently in the CRO team of IVIM Technology with the extensive knowledge of in vivo imaging. She will be based in Boston starting from the end of April. So if you have any inquiries or want to see the system in action, please let us know. We have a demo system in BWH, and we will arrange a demo session for you. And also you can easily RSVP for the online or on-site demos through the link I have just shared with you in the chat box. I will be sharing it throughout the webinar as well. Now let's start with the presentation without further delay. During the presentation, if you have any questions, please do not hesitate to ask in the Q&A box. And now, Hayden, if you are ready, you can go ahead.
Okay. Hello, everyone. Thank you very much for attending today's webinar. I'm Hayden, an application specialist from IVIM CRO Business Development Team. Today, I will present on the in vivo application about cellular level imaging of internal organs in live animals for immunotherapy and cancer research. I have prepared this presentation based on the data we obtained from our previous preclinical CRO service. Okay, the presentation will proceed as follows. First, I will provide a brief overview of an in vivo imaging techniques. Following that, I will present case studies utilizing actual imaging data, including immune cell infiltration and extracapsular spread obtained from animal models using various cancer cells. Let me provide a brief introduction to our company, IVIM Technology. Okay. Uh, First of all, I will start to introduce our techniques first. We specialize in the development of commercially available all-in-one intravital microscopes. Moreover, we offer in-house intravital in vivo imaging services utilizing our accelerated animal facilities. Traditional in vivo microscopes such as X-ray, CT, or MRI imaging were limited to simple whole body imaging or inferring specific organs. In other words, these are macro scale scopes, so it was too difficult to see the mechanisms of the disease development or a drug biodistribution in living cells directly. To overcome this hurdle, we developed the microscale microscope for cellular level visualization, and now we could visualize the drug efficacy and delivery inside of the living body on real time. In addition to the limitations of traditional imaging, researchers had to prepare animal models, imaging equipment, and software for post-processing on their own. However, with IBIM technology, we have optimized a system that is easy to operate and maintains the best conditions for animals. And this allows researchers to easily achieve standardized imaging results, thus increasing the reproducibility in research. The all-in-one single box package enables easy installation, operation, and maintenance. It has co-optimized hardware and software for superb intravital imaging performance with ultra-fast imaging speed up to 100 frames per second. Thereby, we can even capture the fast dynamics such as the red blood cells that are flowing inside of the blood vessels. Together with automatic hassle-free motion compensation functions and various accessories such as a live animal maintenance system and various imaging window system for wide range intravital applications, users easily can get the high quality in vivo data without any limitation in imaging various live organs. Here's the IVIM Technology's key advantages. So we are providing in vivo imaging window chambers that are fully optimized for organ-specific applications, especially for long-term monitoring. For example, with a dorsal skin fold chamber, we could visualize the cells and evaluate drug activities like anti-cancer drugs for after inoculating the cancer cells inside of the skin. And for the lung and the heart imaging, we hold the tissues with negative pressure to reduce the artifacts induced from the breathing and the heartbeats. With our system, all post-processing software is built in. So by utilizing this, researchers can identify details such as the localization of individual cells within the villi of the small intestine, or the nuclei, vascular structures, and more. Furthermore, quantitative analysis in 4D can also be conducted, including the speed and the directionality of immune cell movements as well as the distance between the cells. Okay. Based on the advantages of being able to image organs in a living animal's body at the cellular level, especially thanks to the window chambers, IVIM Technology's current biomicroscope is used for imaging the various disease models, including the cancer models, dementia models, and sepsis models, including the immune cells in lymph node. As you can see, particularly in the left video, we can visualize tumor cells circulating within living animals. In other words, through in vivo microscopy, we can not only observe processes such as proliferations and invasion, but also track immune cell responses and even mechanisms of the metastasis. Using in vivo imaging techniques in living animal models of cancer, we can evaluate the efficacy of various drugs for disease onset and the therapeutic effects. So, I will provide actual installation images of the windows shown earlier as samples. The dorsal skin fold chamber, commonly used in cancer research, is attached to the dorsal skin, allowing easy observation of drug efficacy for cancer treatment and the angiogenesis. The cranial imaging window enables observation of cancer cells in brain tissue, while the abdominal imaging window allows observation of cancer cells in various organs within the abdomen. With just one surgery to insert the window, the advantages of in vivo microscopy become evident as it enables long-term imaging of desired organs. So certainly, let me proceed to the present the result images obtained through in vivo microscopy depicting the growth of cancer cells within tissues and the efficacy of the drugs. For this study, we inserted a dorsal skin fold chamber and injected CT26 GFP colon cancer cells expressing GFP. So imaging was conducted on day 3 and 4 post-injection. Wide area imaging allows us to observe the overall distribution of cancer cells and the vasculature. Additionally, when imaging at high magnifications at the spot 1 and 2, we observed an increase in the number of GLP-positive tumor cells on the day 4. Along with the formation of newly developed blood vessels like this through angiogenesis mechanisms. So here are the 3D videos created through 3D reconstruction post-processing of spot 2 images from day 3 and 4. This demonstrates that IVIM Technology's microscopy not only captures images of cells within the body, but also enables quantitative and qualitative analysis, including area measurement and cell counting, for example. So, these are the result images obtained after injecting the LLC lung carcinoma cells, which express GFP, into the dorsal skin fold chamber model. Red blood cells were labeled with a fluorophore and injected into the mouse, as shown in the video with the green color. This demonstrates that not only can we observe the growth of the cancer cells, but we can also measure the velocity of the blood flow near the cancer tissue. And next, we visualized the delivery process of the pharmaceutical nanoparticles. We injected a triple negative breast cancer cell line into the dorsal skin fold chamber model, followed by the injection of nanoparticles. Subsequently, we conducted imaging at day 2, 6, and 24 hour time points. The nanoparticles labeled in red were observed within the blood vessels surrounding cancer cells at the day 2 and the 6-hour time points. But by the 24-hour time points, we observed the nanoparticles existing in the blood vessels and being delivered around the cancerous tissue. Next, I will present the result data of the efficacy evaluation applying the anti-angiogenic effect of the anti-cancer drug. When administering a drug with anti-angiogenic effects, we observed not only differences in vessel morphology compared to the control group, but also a reduction in blood vessel dilation. As demonstrated, the dorsal skin fold chamber is suitable for long-term imaging of this same location within an individual, making it a very useful technique for analyzing drug responses within the body after administering various treatments. Okay. I will present the results of imaging the process of immune cell infiltration in a breast cancer animals model using the dorsal skin fold chamber. To assess the infiltration of T cells into tumors within the breast cancer animal model, we conducted adaptive cell therapy by injecting fluorescent T cells. Subsequently, we tracked and observed the activation of tumor infiltrating lymphocytes using in vivo microscopy of day 1, 3, 8, and 11. As a result, as shown in the left images, we were able to visualize the activity of tumor-infiltrating lymphocytes for each day. Additionally, through time-lapse imaging on each day, as shown in the right video, we were able to track the infiltration of tumor-infiltrating lymphocytes. Following the previous slide, we further investigated how the activity of tumor-infiltrating lymphocytes differs between the inside and the outside of the tumor cells. As depicted in the left image, the activity level of tumor-infiltrating lymphocytes was higher inside of the tumor compared to outside. This observation was further quantified and graphed, as shown in the graph, indicating a significant and distinct increase in the activity of lymphocytes infiltrating the inner tumor region compared to the outer region. Following the previous slide, where we observed the differences in activity between the inside and outside of the tumor cells, particularly with significant activity inside of the tumor on day 11, we proceeded to observe a xenograft model created by transplanting the human breast cancer cells into mice which were not of the same species. In contrast to the allograft model, in the xenograft model, the strong activity of infiltration was observed on the third day, followed by the gradual decrease in activity thereafter. So, the MDA MB231 breast cancer cell line used earlier was one of the triple negative type. In this study, we sought to confirm whether similar results would be observed using the MCF7 breast cancer cell line, which is a non-triple negative types. As you can see here, in the MCF7 breast cancer model, the activity of the tumor infiltrating lymphocytes was even higher. Therefore, tracking the activity of tumor cells and the tumor infiltrating lymphocytes using in vivo microscopy can be a novel approach to understanding the correlations between immune responses and tumors in living animals. Next, I will move on to the next theme and present the data on the observation of extracapsular spread, the ECS, and metastatic forms of cancer cells in the inguinal lymph node using in vivo microscope. In this study, we directly injected the B16F10 melanoma cell line, which carries the endogenous GFP signal into the inguinal lymph node, and then visualized the process of the tumor formation. So as a result, on the 14th day, melanoma spots within the inguinal lymph node were observed both visually and through fluorescence microscopy. So on the 21st day, with more time elapsed, we could observe even more clearly the formation of the cancer tissue around the blood vessels. On the 28th day, we observed even further, as indicated by these asterisks, that metastasis has occurred. At this point, cancer cells had grown up to the dashed lymph node boundary. You can see here. In this time, we observed the extracapsular spread and metastatic forms of cancer cells in the popliteal lymph node using in vivo microscopy. So in this case, we directly injected the B16F10 melanoma cell line, which carries the GFP signal too, into the popliteal lymph node and visualized the results on the 14th day. At this time, we confirmed tumor formation even in locations close to the boundary of the popliteal lymph node. On the 21st day, when we conducted imaging, we observed the metastasis occurring as cancer cells escaped from the popliteal lymph node, not only inside of the node, but also the outside of the boundary marked by the white dashed line. So on the 28th day, when we conducted wide area imaging of the entire popliteal lymph node, we observed the GFP positive melanoma cells spreading beyond the boundary marked by the white dashed line. So this indicates that extracapsular spread had occurred with cancer cells spreading outside the popliteal lymph node. So we have confirmed the feasibility of tracking the formation process of melanoma in inguinal and the popliteal lymph node. So next, we would like to re-examine whether it is possible to visualize the metastatic process of melanoma step by step using in vivo microscope. Using two-photon laser, we obtained images by visualizing the second harmony generation signals from the capsule of the lymph node as indicated by the dashed line marking the boundary of the popliteal lymph node. So additionally, we stain the blood vessels to visualize them along with the cancer cells. So as a result, although there were individual variations, we observed and visualized all steps from the stage 1, where the melanoma grew near the boundary of the lymph node, to stage 2 and 2.5, where it grows where it grew close to the boundary, and finally to stage 3, where the extracapsular spread occurred as the melanoma escaped the capsule of the popliteal lymph node. So, in summary, I like to emphasize again that with the use of in vivo microscope, it is possible to longitudinal track the growth process of tumors as well as their interactions with the surrounding tissues, leading to metastasis all within the single animal subject. So using IVIM Technology's in vivo microscope, we continuously optimize the methods for imaging a wide range of small animals, including mice, rats, and ferrets, allowing for the unrestricted imaging of multiple organs. This not only extends to the visualization of cancer animal model using the dorsal skin fold chamber, but also encompasses various other applications. So based on this, IVM Technology also offers the research services. We engaged in discussions regarding the experimental and application aspects of using in vivo microscope in ongoing research. Following this, we provide a one-stop total solution research service encompassing post-processing and both qualitative and quantitative analysis of the data. IVIN Technology is committed to ensuring that researchers obtain the meaningful results in cancer research, spanning all organs from the brain to the prostate and beyond. So thank you for listening attentively for a long time. So if you have any questions, please feel free to ask.
Thank you, Hayden, for the nice presentation. Now we will be answering your questions. We will also answer the questions previously asked through the registration. The first question is from Rick, and he is asking what depth of tissue can be imaged. Also, is developing a kit for imaging a new organ, for example, spleen, readily doable.
Okay, thank you for the questions, and I'll let me say that the imaging depth varies depending on the tissue or disease model of interest. But typically, confocal microscopy can image up to 100 to 300 micrometers, whereas two-photon microscopy can reach around 500 micrometers. And also, there are specialized and optimized imaging chambers for visualizing the spleen. As I shown in the previous slide, the abdominal imaging windows could be successfully used for the imaging the spleen for a long term, like 4 to 6 weeks.
So, another question is, for in vivo monitoring, are the T cells and B cells prelabeled by genetic approaches? Can you stain various immune cells with injected fluorescent antibodies for imaging?
For the fluorescent visualization of the target cells, uh, you can use the transgenic mouse or the genetically modified cell lines or other things. But in the case of you have to use the wild type mouse or the normal cells and the immune cells, we optimize the in vivo labeling kit for visualizing the T and the B cells, which we can use the antibodies targeted to specific epitope which the T cells and the B cells has the special epitope, so we can use that. So we can inject the in vivo labeling kit targeting the specific epitope to visualize the different colors or for using the different wavelengths for the our in vivo microscope system. I hope that would be the good answers for you.
I think it was good. Thank you,
Thank you.
Hayden. And if you guys have any other questions, please do not hesitate to ask in the Q&A box. Meanwhile, I will be asking the questions previously asked. So They were asking for long-term observation, how many days, up to how many days can you typically image for?
Okay. In the case of the dorsal skin fluid chamber, we can maintain imaging for approximately 2 to 3 weeks. And in the case of the abdominal chamber used for the abdominal organs, such as the spleen and the pancreas, liver, it can sustain imaging for 4 to 6 weeks. And additionally, researchers studying the brain tumors can utilize the cranial imaging window implant for imaging of the brain for over a year. So this is very good tool for studying the brain tumors and also as well as the Alzheimer's disease or as other diseases.
Mhm. So the other question, I think the answer will be kind of long, but
Yeah.
if you can give a brief summary. So which kind of data can be acquired using your two-photon microscopy, not the confocal? Like what's the difference? What can I image through the two-photon but not the confocal?
Okay. I cannot simplify the answers because this is a very important question. So typically, tissues with repetitive structures such as the collagen and the fibers and the muscles exhibit the second harmonic generation signals, eliminating the need for fluorescence labeling. So therefore, we utilize two-photon imaging to visualize, for example, the boundary of the lymph node capsule using second harmonic generations. And also, if fluorophores compatible with two-photon microscopy, such as Evans Blue or the Cetal 647 are available, two-photon fluorescence-based in vivo imaging is feasible. So two-photon microscopy offers the advantages such as reduced photobleaching and a deeper imaging capability, while confocal microscopy supports a wider range of the dye types. So researchers choose between them based on their experimental design.
Mm-hmm. Thank you. And if you guys have any other questions, please do not hesitate to ask now, or you can send through email. I think uh, there are no. Oh, okay, there's another question. So one more. Okay.
Okay.
We are waiting for your break.
I like to tell you that in the case of the fluorescent labeled wild-type mice, we produce and also sell the customized in vivo labeling kits called the IVI Tech. This antibody-based kit specifically binds to the epitope of the target cells, minimizing the non-specific fluorescence signals and enabling the specific labeling of the desired cells at desired wavelengths.
I think this answers a couple questions previously asked.
Yeah.
And Rick is also asking for long-term imaging, does the specimen need to stay attached to the instrument?
Yeah, sure, because the implantation of the imaging chamber is for the long-term imaging with one surgery. So it has to maintain with the this imaging chamber implanted on their bodies. But I think they are not that feeling uncomfortable with that because after 1 or 2 days for the recovery, they can move or they can move and eat well, sleeps well. So mice can be maintained healthy with these
Yeah.
abdominal or other imaging chambers.
Yes, you mean after the surgery, after the imaging, and that it can live its in own cage um,
Yeah, sure.
for up to a couple weeks, and we can utilize the same animal model again for the imaging. So we don't have to
Yes.
sacrifice the animal, right?
Yes, and also it enables this same spot imaging because we use the one individuals for this experiment. So that's the another huge advantages as well.
Yes,
Yeah.
that was a great answer. Thank you. And thank you everyone for your questions and also your active engagement. Before we wrap up, I just have a special announcement about the intravital imaging workshops that we are having right now in Boston at Brigham and Women's Hospital. So it is planned for Hayden. It's planned for the last week of April. all of the bookings until April is all full, unfortunately, and we have only a few slots left for the April and May as well. So if you are interested, please hurry up and RSVP through the link that I have shared in the chat box. And also, if you are interested, you can send through the email information at ivimtech.com. And thank you again for being a part of this webinar today, and we will see you next time. Thank you.
Thank you, everyone.